Hello, good morning students. Welcome to my video. And today we'll be talking about the structures of chromosomes and the DNA here in the class. Before we start with our topics here, I want you people to have a look at these different 3D diagrams of chromosomes and the DNA. These are different chromosomes, and these are the DNA. And then how within the DNA we find the genetic information, the genes, which carries the genetic information from parents to the offsprings. And then before we start with the topics here, the chromosomes and the structures of DNA, I want you people to give a thought over a numbers of important questions. These questions are, number one is we will discussing in this class, we'll be discussing what the first is, what is chromosome. So please give a thought over what is chromosome. Second important question is, when can we see the chromosome in the cell? Third, where do we actually find the chromosome? And how many chromosomes do we find in the diploid cells and the structures of the chromosomes and the DNA? So in our classes, we will be discussing these 5 different questions here. So let's go back and first discuss about what is chromosomes. When we see the chromosome, this is your cell. We find the chromosomes here in the largest cell organelle called the nucleus. So this is the nucleus, and rest of these are different cell organelles. You can see this is the mitochondria, this is the cell membrane, and this is the largest cell organelles. In this largest cell organelles, we find the chromosome. But here, it's written here chromatin. What does chromatin mean? That means if the cell is at rest, suppose if this cell is at rest, you can see the chromosome only if the cell is in the process of cell division. Suppose if this cell is at rest, you cannot see the chromosome. The chromosomes are always decondensed and forms the chromatin fiber, and they remain within the resting cells. We learn that the resting phase of a cell is the interphase. We have discussed about all kinds of subphases of interphases. But the same cells, once they start to divide, once the cell enters into the different phases of the cell division, this cell slowly, inside the cells here in the nucleus, the chromatin fiber slowly condense and forms the chromosomes. So chromosomes are visible only during cell divisions. And then we know that the chromosome was first discovered by the German scientist called the Wettler Fleming here in 1882. And then literally the chromosome means chroma. It comes from the chroma, which means the colors, and soma means the body. Hence the name of the chromosome means, or literally the chromosome means colored body. Okay, so here, thus in the largest cell organelles, we find the chromosome. So how many chromosomes do we find in the nucleus? Look at here. We have, in case of human beings, we have 46 chromosomes are found in the nucleus. We know that these are autosomes and these are the 2 sex chromosomes here. So this 46 chromosomes, and these 46 chromosomes are always found in the pairs. That's why we call it 23 pairs. Look at here, first pair, second pair, third pair, fourth pair, fifth pair. It goes on, and 23rd pairs. So this chromosome, 46 chromosomes are always found in the pairs. That's why we call it 23 pairs. So these 23 pairs chromosomes are found in the nucleus of every diploid cells of human beings. Every cells of your body contains these 46 chromosomes. Okay, so now let's discuss about the structures of the chromosomes. If you discuss about the chromosomes here, the structures of a chromosome, look at here, the chromosome has 2 important arms we can see. These arms of the chromosomes are called the chromatid. And these chromatids are attached to a point called the centromere. So centromere, sometimes in your questions, in the competitive question, as well as in some medical question, as well as even the ICS and CBSE, they ask a question like, where do we find the centromere? The centromeres are found in between the 2 arms of chromosomes, okay, or between the 2 chromatids. So locations of centromere is very important. And what does the centromere do? The centromere, as I've written here, it serves to attached to the spindle fiber. So in the process of cell division, since we have already done the different process of the karyokinesis, or the phases of karyokinesis: prophase, metaphase, anaphase, and telophase. In these phases, we learn that the spindle fibers are formed, and then the separations of the centromere occurs, okay, due to the spindle fiber. So spindle fiber actually attaches to the centromere of the chromosomes. So this is the functions of the centromere. Then if you see these two arms of the chromosomes are chromatid. Chromatid is attached to a point called the centromere. So each chromatid is actually made of what? Chromatin fiber. So chromatin fiber. You can see the chromosome. Each of these chromosomes has a 4 arms, altogether 4 arms. This kind of chromosomes are called duplicated chromosomes. So sometimes you may be asked to draw the duplicated chromosomes also in your ICC exams. So make sure when you are asked to draw the duplicated chromosome, you will draw this kind of diagram. Okay, chromosomes with 4 arms are called duplicated chromosomes, and then 4 arms and attached to a point called the centromere. Now, these chromosomes, as I told you, is nothing. It's simply made of a chromatin fiber. So if you go back and see the chromatin fiber, look at here. This is your chromatin fiber. So this chromatin fiber slowly condenses and forms the chromosomes. So here is the chromosome. Chromosome is nothing. It's made of what? Chromatin fiber. But the question is here, we will be discussing about the chromatin fiber here. So what do we actually find in the chromatin fiber? If you see the chromatin fiber is actually made of 2 important substances. You can see the chromatin fiber is made of 2 important substances. One, a long string-like structures here. This string-like structures is the DNA, and then oval round proteinous structures here. These proteinous oval structures are actually, they are the histones. So histones and then DNA. So your chromatin fiber is made of 2 important substances. One is the histones, which is the positive charge, whereas the DNA is the negative charge. So negative and positive charge, they stick together, they remain together in the forms of nucleosome. We'll come to know about and discuss about the nucleosomes in our classes here. So this is your DNA and histones. Now if you see this chromatin fiber, almost 60% of this chromatin fiber is made of histones, these histones. Whereas 40% of your chromatin fiber chromatin is made of DNA. And this DNA actually, which carries the genetic information, which is called the gene. We'll discuss about the gene and then DNA in details in our classes here. So now look at here. When you see the DNA and histone here, this is another diagrams of the chromatin fiber. This DNA and histone, they remain together because of the force of attractions is built between the DNA and histones. As I told you, the DNA is the negative charge and then your histones are the positive charge. Now, this kind of a complex structures, if you just count the numbers of histone here, number 1, 2, 3, 4, 5, 6, 7, 8. All 8 histones, and on the surface of these 8 histones, the DNA winds around it. Okay, here we see the 8 histone, and then DNA winds around it. A core of 8 histones, and then DNA winds around 8, is called the nucleosome. So this kind of a complex structures which is built between the DNA and histone is called the nucleosome. Just keep that in your mind. A numbers of questions are asked in your ICC exams. Okay, then let's talk about now. Forget about these histones. I told you about the histone. They are the proteins in natures, and then they are positive charge, whereas DNA is the negative charge, and we will discuss about the DNA here in our next classes. Now, when you see the DNA, look at here, you have, this is the DNA. So the DNA is one of the largest molecule found in the cell. Among a number of largest molecule, one of the largest molecules in the cell is the DNA. And since it is one of the largest molecule, we call it the macromolecule. You will come across these terms here in the classes, macromolecule. So DNA is one of the very large single molecule, biomolecule, that's why it is called the macromolecule. Now when you see the DNA, look at here, please we'll move down here. If you see the DNA is exactly, you must have seen the spiral staircase. DNA is just like a spiral staircase. Now, if you just stretch this spiral staircase, you'll find a ladder-like arrangement here of a DNA molecule. So this strand DNA has two important parallel lines here. You can see the two parallel lines. One line runs down here, this line runs here. So these two parallels of the DNA is called the strand. S-T-R-A-N-D, strand. Two strands of DNA are here. These are two strands of DNA. But with the help of the powerful microscope, today we can even see each strands of DNA also. So look at here. When you see each strands of DNA, the DNA, each strands of DNA molecule is made of repeating nucleotide. We call it repeating nucleotide. Now, one single nucleotide has three different components. The first component is called the phosphate. Second component is the ribose here, pentose sugar. Pentose means 5 carbons here, pentose sugar. And then base here, the nitrogen base. So this is one nucleotide. So one phosphate, one sugar, one nitrogen base combines together and forms one nucleotide. Another here, phosphate, sugar, nitrogen base, one nucleotide. Phosphate, sugar, nitrogen base. And on the other side also, if you see a phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base. So what we find here is each strand of DNA is made of what? The repeating nucleotide. So DNA molecule is nothing, is simply made of what? Repeating nucleotide. And then I told you in each nucleotide there are three different components, phosphate, sugar and nitrogen base. And then we know that phosphate and sugar, they are arranged lengthwise, whereas the nitrogen base is inward, which joins with the nitrogen base of other nucleotide. In between these nitrogen base, we find the hydrogen bond. Now, how many nitrogen base? Now look at here, the nitrogen base. If you just talk about this nitrogen base, how many nitrogen base do we see here? We see 4 different types of nitrogen base. Number A, T, and GC. 4 nitrogen base. AT, you can just remember Ansel Thapa or Anit Thapa. I think it's very famous in Kursium. Okay. A stands for adenine. T stands for thymine. GC stands for Ganesh Chetri. You can write Ganesh Chetri. Just remember Ganesh Chetri. But G stands for guanine and cytosine. And then in between adenine and thymine, we find the double hydrogen bond. Whereas in between guanine and cytosine, we find the triple hydrogen bond. These hydrogen bonds are the weak bonds. Okay, there is an enzyme called helicase enzyme which can run through this hydrogen bond and can separate the two DNA strand. We can discuss about that. And then hope you understood these particular topics here, the strands of DNA. And if you just move down, how this DNA replications of the DNA here. Okay, replication of DNA is also called the copying of DNA. It's also called the duplications of DNA. How? From one single DNA molecule, 2 different molecules are formed. We learned that, okay, in case of the mitosis cell division, the number of chromosomes or the DNA molecule doesn't change even in the daughter cells. It forms a 2 daughter cell in the mitosis cell division. 46 chromosome forms, 46 chromosome in a diploid cell divides and form the two daughter cells, and each two daughter cells will have the equal number of chromosomes, that is 46, 40. That is possible only because just before the cell divides and forms the daughter cell, the DNA replication occurs, duplication of DNA occurs, in which one of the single molecule of DNA forms the two molecules of DNA here. So how does this DNA replicate? So here is one single DNA molecule. Now you can see these two strands, and we learned that two strands of DNA molecule is made of what? The repeating nucleotide. We have already discussed about that in details. Now, these in between the rungs of this DNA, if you just see the rungs of the DNA, you can see here, or rungs of this ladder. These rungs of the ladders are made up for the nitrogen base. And then in between the nitrogen base, we find the hydrogen bond. As I already told you in the previous, in this class only, that these hydrogen bonds are a weak bond. And then there is an enzyme called the helicase enzyme which can run through it and then separates the two strands of DNA. So this is what we exactly see here. So helicase enzyme has separated the two strands of DNA, and then a new strands of DNA is formed here. You can see new strands of DNA is formed here. And then eventually from one single DNA molecule, two DNA molecule is formed in the replication of DNA. Okay, so this is all about the structure of chromosome. But before I end here, I just wanted to revise once again quickly. It's very easy to understand the DNA and chromosome. Simply what you need to know is think about a cell. The cell has the largest nucleus. In the nucleus we find the chromosome. The chromosomes are, as I told you, visible only during the process of cell division. In the prophase, metaphase and anaphase, we learn that. And then once the cell is in the process of cell division, the chromosomes are visible. And then the chromosomes, these chromosomes have two arms. These arms of the chromosomes, we can go back and I just wanted to show you. The arms of the chromosomes are a chromatid. The arms of the chromosomes are called the chromatid. Okay, these arms of the chromosomes are called the chromatid here. Chromatid, and these chromatids are attached to a point called the centromeres. And then each chromosomes are made of what? Chromatin fiber. Chromosomes are nothing, it's simply made of what? Chromatin fiber, I told you. So chromatin fiber, when you see the chromatin fiber with the help of today's technology with a high powerful microscope, we find two important substances that compose or that is present in the chromatin, the histones and then DNA. So we learned that there will be a formations of complex structures between the histones and DNA is called the nucleosome. We discussed about that. And then when you see the DNA molecule, this DNA molecule is like a spiral staircase. When you stretch them and make them, straight, you find just like a ladder. And then two strands of DNA. Okay, this DNA has two strands, and each strand of DNA is made of what? Repeating nucleotide. Nucleotide has got three different components. We discussed about that: phosphate, sugar, and nitrogen base. We have altogether 4 different nitrogen base. In simple word, we can remember them in adenine, AT, and GC. I told you to remember that. A stands for the adenine and adenine, and T stands for thymine, G stands for guanine and cytosine, and there is a weak bond, hydrogen bond in between adenine and thymine. There are double bond, whereas in between G and C, that's guanine and cytosine, we find the triple hydrogen bond. And how this DNA molecule replicates in the separations of two strands of DNA occurs, and then how from one single DNA molecule to DNA molecule is formed. And then the last topics we can see here, go back. If you just go back, you need to know what is actually, please go back. Yeah, if you just see this diagram here, okay, this is the diagram. What is actually a gene? That this gene here, which actually carries the genetic information. What is genetic information? Is simply the sequence of nitrogen base. So these different parts of the DNA which has an information to produce the proteins are called the gene actually. The part of the DNA or the segment of a DNA which has a genetic information to produce the proteins are called the genes. And genes are the hereditary characters or which carries the hereditary characters or which controls the formations of the protein in our body. Thank you so much, students. We'll see you in the next video. Thank you so much.